webpage. So mitosis and meiosis, you have these pages out and ready to go. Take a look at the screen. This is under genetics for Unit 3. This is outcome 2. It says mitosis and meiosis. And let's start with mitosis. So here is notes that I posted in class from a previous year. So we want this ready to go as a reference. Now, would you like me to talk through these parts here and then go back to the first page, or do you want to deal with the fill in the blank first? Is there a preference? This fill in the blank, just the other parts first. Okay, let's talk about mitosis. When we speak to mitosis and meiosis, how would you summarize what those processes are about? Correct. Both of them are about cell division. Mitosis, which we're going to start with, where does mitosis happen? If we're talking in the human body, where does mitosis happen? Good. Do we know what somatic cells are? Let's quickly Google what somatic cells are. And remember, you don't have to stress about having everything perfect because you have a reference to go back to here as well online. So let's Google what somatic cells are, and then let's do hands up once we've found an explanation for somatic. I believe that's S-O-M-T-I-C. So M-A-T-I-C, sorry. Somatic. Or I guess the opposite of somatic cells would be Mitchell.
Is any biological cell forming the body of the organism?
Good. So any type of cell that's found in an organism, we would talk about mitosis basically happening in anywhere in the body. The only place it isn't really the main type of cell division is what location? Yes, in the reproductive organs. Meiosis happens in the reproductive organs because we want to cut the chromosome number in half, so it's a different type of cell division. So what you want to note about mitosis happens in all cells of the body. It's what happens in plants. Basically anything that is getting larger, taller, wider, or if cells are being repaired, it's because of mitosis. Now, if you were to look inside of a cell, so if this is our cell, where in that cell would mitosis take place? Correct. So it's happening in the nucleus. And what's inside of the nucleus that we care about? Right. So it's the DNA inside of the nucleus. We learned that DNA comes in those long strands. What are those long strands formed into that we care about in genetics? Right. So what we really want to know about is chromosomes. And in the human body, how many chromosomes do we have in each nucleus? We talked about it in class before. How many chromosomes in the human body? Correct. So 46 chromosomes. Every cell in your body should have 46 chromosomes. If you had 47 chromosomes, what does that mean? Could mean Down syndrome if it happened at the 21st pairing. Could mean Edwards syndrome if it happened at another numbered spot. So anything above or below this number has major consequences for a person. And most often you won't survive except in select genetic disorders like we were talking about. So we know what happens in cells, in the nucleus. Then what we have to think about is the cell cycle. Most of the lifespan of a cell is spent in what's called interphase. So if we were to draw out interphase, where am I going to put this? So interphase, if we made it kind of like a clock, most of the cell's life would be spent over here. This is interphase. So most of the time is spent in G1, S, and G2. G1 is a growth phase, so the chromosomes are, or the cell is growing bigger. The organelles that are in the outside of the nucleus are copying themselves. Then we hit S, or the synthesis phase. That's where the DNA replicates. So you remember back in Unit 1, we lined up, we made a copy of DNA right out here. That's how you make copies of the chromosome. So after the S stage of their life cycle, there's now double-stranded chromosomes versus a single-stranded chromosome. So what that means is, in G1, that's how we would draw a chromosome. It's the rest of the cell that's getting bigger. There's more mitochondria being copied. The Golgi apparatus are being copied. After the S phase, then we have double-stranded chromosomes. So this side is an exact copy. So this side is an exact copy of that side. Okay, and the reason that we do that is so that we can split them in future cells. So this is what they look like in G1. This is how they end in G2. If G1 is a growth phase and the cell is getting bigger, then we copy the chromosomes. What do you think happens in G2? G2 would be growth, the second growth phase. The cell reaches its kind of a maximum size. It finishes copying any organelles that need to be done. What I'm trying to show is time. Most of the time is spent in G1, S, and G2. So only a very small portion of this clock here is actually connected to mitosis and cytokinesis. So your cells aren't dividing all the time. They go through this normal phase where they get bigger, they copy the information. When it's time for them to divide, you're growing as a person, you need more cells. That's what they do. So that's a small portion of that time. When we reach that stage, we move from interphase, which is the G1, the S, and the G2, into mitosis. What is the first stage of mitosis? Right, so that's where we are going to begin, is with prophase. Now, in the example that I have here, how many chromosomes have I drawn? Two double-stranded chromosomes, right? They're ready to divide. So here we have 2 chromosomes. Important to note that it's 2 chromosomes there because we want to check the number of chromosomes at the end. If mitosis is happening in all cells of the body and I start with 2 chromosomes, in the cells that are produced from that, how many chromosomes would you expect? If we're just dealing with 2. If I started with 2, you'd want me to go to 92 chromosomes? If I'm making new cells, so I'm making more cells, how many chromosomes do I want the new cells that I make to have? In humans, it would be 46, right? We want every set of chromosomes, or every cell to have 46 chromosomes. In this organism, if I started with 2, I would end with 2. So in mitosis, whatever you start with for chromosomes is what you end with. So it's an exact copy. So in prophase, prophase being the first step, This is something where you have to learn the steps in order. You cannot rearrange the steps. University professors will not find that funny. You have to make sure you learn it prophase, metaphase, anaphase, telophase. So prophase being the first one. What are some key things that you found that happen in stage 1? Pardon me? Excellent. The nuclear membrane that surrounds it dissolves. So one of the things you should have mentioned here is the nuclear membrane, which is this part out here, will dissolve. Now it's just the chromosomes inside of the cell. So nuclear membrane dissolves. The other thing that you have forming are what are called spindles. So you have the formation of spindles. Spindles are these little lines that reach out from the centrioles. Remember we learned about centrioles in the last unit. Reaches out and grabs onto the centromere. Centromere is like the middle part of the chromosome. I would say that centrioles are like Spider-Man. And in this classroom, if you were all paired up with one other person and linked together, Mitchell would be the other Spider-Man on the other side of the room, and we'd be able to shoot webs out, and we would attach one of those webs from each of us to each chromosome at the central part. So from the centriole, we send out this spindle, which attaches to each side of the chromosome. The other thing that happens is that you can see the chromosomes at this point. And what we will try to do is find some websites where you can actually see what the chromosomes look like in those early stages. So it's the point when you can actually see them inside of the nucleus. So prophase, no nuclear membrane, spindles form, we can see the chromosomes. Any questions? You're all being extra quiet today. Next phase is metaphase. I always remember that metaphase has to line up in the chromosomes always line up in the middle. M for metaphase, M for middle. So metaphase, the spindles pull from each side until they line them up in the middle. So here you have a chromosome lined up in the middle because these the spindles are pulling it to get there. They do not line up in pairs. They line up one above each other, all connected with the spindles. So metaphase, line up in the middle. Are we okay with that? Nodding or shaking your head helps me at this point. Anaphase. Next stage is anaphase. And what I've tried to show here is if we had two people linked together and a spindle attached on either side, those spindles start to recoil. So Spider-Man's pulling them in. So essentially what happens is they split. Where it was double-stranded, they now split and become single-stranded. And if I was one strand of that chromosome, I get pulled over to this side. They often talk about pulled to the poles. So I get pulled to this side. So I still have all the information that my partner chromosome would have had, because it was just a copy, right? We did DNA replication to get that copy. I now get pulled to that side. So anaphase means they get split apart and pulled to the opposite side. Are we okay? Telophase is when you have the cell membrane is still here, but now you have the nuclear membrane reforming inside of the cell around those chromosomes. So if I went to this side, anybody else that had been pulled apart, we would be surrounded by the nuclear membrane here. Same thing is happening over there. That's called telophase. The next step is called cytokinesis. That's the very last part there. So telophase, if you wanted to make a note there, telophase is when the nuclear membrane reforms around those chromosomes. So the nuclear membrane reforms around each set. There's now 2 nuclei in one cell. Then basically what happens is you take this cell that had the 2 sets of chromosomes in it, and these sides start to move inward. And cytokinesis, which means dividing of the cytoplasm, then it starts to look like this, and then it starts to look like this, and then eventually you get 2 cells that are side by side. So if we all joined hands in one big giant circle, and I said Matt was on one side and he's going to move to the middle and I'm going to walk to the middle until we get together, then that would mean that we would have created a cell on this side and a cell on this side. So the cytokinesis is when the cell membrane comes in until it joins. Now, what about in a plant cell? Can the cell membrane in a plant cell just go to the middle?
No.
What do plants have that make that difficult? Pardon me? Right, there's a cell wall in place. So what I was trying to show in this example here is that first this is all inside of the plant cell. Then what you have in the next situation is a little growth plate appears. And that little plate keeps getting bigger and bigger till it touches on either side. And now you have 2 cells. And then those cells would get a little bit bigger as they grow, and they push everything out around them. A couple really important things to remember when you deal with mitosis. We started with 1 cell. We ended with how many cells? 2. So we always essentially go from 1 cell to 2 cell. So all of us started out as single cells. That cell divided and formed 2 cells. Then the 2 cells formed 4 cells, and 4 cells formed 8 cells, and so on and so forth until you are thousands, if not millions, of cells in your lifetime. So mitosis is fairly significant to us. We started with, in this case, we drew out 2 chromosomes. So at the beginning, inside of the cell, we had 2 chromosomes, right? At the end, you have a cell that looks like this. Does it still have 2 chromosomes? It's just in the G1 stage, so it hasn't duplicated itself. So it doesn't have a second strand. So in the end, you start with 2 and you end with 2. If you go through mitosis and you don't end up with the same number of chromosomes, that's when it causes problems. Now, when you deal with cancerous cells, remember I drew that little clock where most of the time is spent growing, copying the DNA, growing some more? Cancer cells short-circuit that cell cycle. And when they go through mitosis, they rapidly complete the cell cycle, so they are ready to divide in a matter of days, hours, months, depending on the type of cancer that you're dealing with. So you want to make sure that they're always following that proper life cycle. If they're not, then there's problems that will arise from that. Questions that you have about mitosis? Okay. Then let's pause for a moment and switch our brains into meiosis, and then we'll use that fill in the blank page as a check-in. Okay, this is meiosis. Earlier we said meiosis happens in which cells of the body? Sex cells. So meiosis, so close, happens in the reproductive cells of the body. Let's refocus. The point was to get some energy, not to lose track of you all together. Turn to a partner, and when I say go, you are going to name the 2 types of sex cells. Go. Incorrect.
X,
Yes.
X and Y.
It has to do with the X's and the Y's. But what are the two types of cells? Egg cells would only have what type of chromosome? X, and sperm cells could have X or Y. So it depends which sperm cell fertilizes the egg as to whether the baby is male or female. Which is why it's ironic that King Henry VIII killed all of his wives for having girls.
It was his fault.
It was. So, meiosis happens in the reproductive cells. If, gentlemen, there are 46, ladies and gentlemen, we need to refocus here. There's a couple key differences off the start that you need to note. If meiosis is happening in the sex cells, and humans have 46 chromosomes, how many chromosomes are in an egg cell?
23.
Correct. So we need to cut the number in half. Meiosis is the type of cell division that reduces the chromosome number to half. So that's what you have to remember. The whole purpose here is to go from 46 to 23. We, however, won't draw out 46 chromosomes because that would take forever. So how many chromosomes are in this stage? No. How many have I drawn on the screen? If this is one chromosome, how many more are there? I'll come back to that. Just don't let me forget. If this is one chromosome, how many chromosomes do we have, ladies and gentlemen? Correct. So this is 1, 2, 3, 4. Now, did any of you come across anything that said something about homologous pairs? I want you to Google homologous pairs. So homologous pairs . What's a homologous pair? Very subtle. Switch. Homologous chromosomes are, ladies and gentlemen, what does it say, ladies? Homologous chromosomes? Your phone works, though. Yes. Mitchell, what did we find?
Our chromosome pairs were crossed from the same length, center, mirror position, and dating pattern of the genes to the same period as the corresponding rows.
So, homologous chromosomes, ones that are similar in size, similar in shape, and gene arrangement. So, a homologous chromosome needs to be one that matches up and has the complementary information. We said everything occurs in pairs, so you would either have dominant on one and recessive on another, or dominant and dominant, or recessive and recessive. That's what's going to create the variety. So, a homologous pair. I want to, for just a moment, draw out what happens with homologous pairs. And we need to go to the front page at the bottom where it says draw an example of crossover. That's what we are going to do. I'm just going to get a clear screen here. So this is crossover in homologous chromosomes. Ooh, I got a little crazy. So, get on the bus, keep your arms and legs inside the vehicle, pay attention. we need two colors, or you need a pen and a pencil to draw this out. Chromosome number 1 is going to be red. Chromosome number 2 is going to be green. Then you could use blue and red. You could use pen and pencil, as long as you have two colors. Yes, or anywhere on that page. Actually, I think I said on the front page, and we could check the video to see. So, because they are similar in size, shape, and gene arrangement, they pair up together. The important part of that is that crossover occurs. Crossover is what creates genetic diversity and variation. So, on this chromosome, let's say we had the dominant trait for both gene A and gene B. On this chromosome, we're going to have the recessive trait, just as an example here. And now, essentially what crossover means is if I was standing beside someone and I could detach my arm and reattach to them and take their arm, that's what these chromosomes are going to do. They are going to switch arms. They are going to cross over to the other chromosome. I want you to think about the impact that has on the genetic information. So after they go through the crossover event, they will look like this. And where we would have had big A stays the same. Does little a stay the same? Did we flip the top ones? So then they stay the same? I had a big B with the red. You'll want to finish this out before you go. So big B stays with the red. Little b was on the green. And now when you look at the types of gametes that will result, here the only option was a big A and a big B. Here the only option was a little a and a little b. Now this one codes for big A, big B. This one codes for big A, little b. This one will code for little a, big B. And this one codes for little a, little b. So it creates 4 options for the gametes rather than just 2. So crossover is very important for genetic diversity. There is also a thing called double crossover where they switch and then they switch back that you'll learn about in a genetics class at university. But crossover basically means these 2 parts switch places. And once you have that down, you have a wonderful weekend. Monday we will finish up with meiosis. And if you need to review incomplete and codominance, do that. Study sessions on Tuesday and Thursday.